Yeah, you can go back. Yeah. So that's how you tell it. Yeah. Okay, let's see what question class. What are you? All right, 10 minutes, answer these 5 questions and submit. Okay. Okay. All right, 7 more minutes. Guys, focus and answer these 5 questions. Put your cell phones away. You don't need those. Put your cell phone away. Answer 5 questions and turn them in. It's right here, man. Okay, you guys have 4 more minutes. Okay, you should be turning it in. All right. Everybody should turn in their warm up. All right, so let's do this together. What is this process that you see up here? What is that picture? Transcription. Now you know it's transcription because look, I've got a DNA molecule. Look, it's opening up. RNA polymerase does everything, does the whole job. Unwinds, unzips DNA. And then I can see a single-stranded mRNA molecule being built. I know it's mRNA because A, it's single-stranded, and B, there's a whole bunch of U's in there. Okay, so I know that it is an mRNA strand of mRNA strand there. 3 ways that RNA is different from DNA. First one, somebody tell me. Very good. RNA has ribose, sugar, and DNA has deoxyribose, sugar. Good. One.
Is it cool if I just like put RNA and DNA and like just these on that too?
Well, I don't know. Tell me what you said. Tell me what you wrote.
Alright, for RNA, I put U and A match together.
Okay, so RNA does not have thymine, but has uracil. What's another one? Third difference. Good. DNA is double-stranded and RNA is what? Good. That is straight out of your notes. Straight out of the notes. All right. Here's a DNA strand here, GCT, AAT, CCG. Now it's asking you to transcribe that DNA into RNA. So starting with the first 3, GCT. What would be the RNA complement to GCT? CGA. I should hear more than just one person, guys.
CGA.
Hold on. And so now AAT, you guys all together. UUA. Because I know that RNA, oops, 1, 2, 5. I know that RNA doesn't have T, right? So it's going to be a U. So UUA. All right, and then last 3, CCG. CCG is what? GGC. Good. So that is the RNA or the mRNA complement to that DNA. Okay, you could have seen that up here. So notice how on DNA there's an A, and on RNA there is no T, so it's U. And then G is C, and then T, A, C, G, A, then U, C, then G, and so on and so forth. So that was kind of the clue there. All right, in RNA, diamine is replaced by what? Good, uracil. In eukaryotic cells, and we say eukaryotic because eukaryotic cells are the only ones that have this particular part, cell part. Transcription takes place in the what? What part of the cell does transcription take place?
Polymerase?
No. What organelle? Only eukaryotes have it. The nucleus. Okay. Please correct yourself in a different color ink and post your corrections, turn in your corrections. When you're done with that, after you turn in your corrections, go ahead and click on the notes for today and set up your notebook. So it says protein synthesis notes for today after you turn in your warm-up. Make sure you set up page 1 and page 2. Please do that now. Set up page 1 and page 2. Please do that now. You mean protein synthesis? Right? Protein synthesis part 2. Yeah, not photosynthesis. All right. Please do that now. That is what we are doing. Please set up your notes for protein synthesis part 2, page 1 and page 2. Please do that now. Hey, Carlos, you should probably put your cell phone away.
Huh?
Put your cell phone away and start writing then. Okay, open up your Chromebook and get your notes set up. All right, for those of you at home, I'm going to go over how to use the codon chart, but I'm going to use my my document camera. And unfortunately, I can't display for the students here and for you at the same time because document camera won't do that. So you will have to look at the video. It says how to use a codon chart. So you can, always mute me when I say that we're about to practice in the codon chart. You can mute me and then just watch the video so that you can actually see what it is that I'm doing. Because I know you can't see it when I show it to them. Do you guys understand that? Yes, no, maybe so. Can you guys hear me?
Yes, ma'am. Yes, ma'am.
Okay, good. All right, so that's the only time. So if you hear me say that we're going to go to the document camera and write this down, just know that the video is there for you to watch because you won't be able to see it on Teams. All right, so bear in mind if I go too fast, you guys have a copy of these notes on Canvas. So I don't mind if I am going too fast. Okay. All right, just what I wanted to do, I want to start off by kind of reviewing what we talked about last time, just to make sure that everybody kind of is on the same page before we move on. So if you would go back to part one, so look at your part one notes, protein synthesis part one. Oh no, we're not doing that. We started with transcription. Over what DNA is. We said that DNA is in the nucleus. DNA can never leave the nucleus because that's where it's protected. And in fact, the more melanin you have, the more protection you have for your DNA. We said that proteins were chains of amino acids. Amino acids are their monomers, so it's kind of like building blocks. And so the purpose of DNA is that it is the instructions for building protein. But there remains an issue. because the DNA is locked in the nucleus, but the proteins are built out in the cytoplasm. So we've got to get the information from DNA in the nucleus out to the cytoplasm so the proteins can be built. So what we do is we copy that DNA into a molecule of what we call messenger RNA. And that's transcription. So in the nucleus, we're going to copy the information from DNA into a molecule of messenger RNA. And then the messenger RNA is going to leave the nucleus and go out to the cytoplasm, where it's going to hook up with the ribosome and start to actually build the protein. So today, we're going to talk about how this process occurs. We went through this, so I don't have to do that again. So we said that transcription was the making of mRNA from DNA. So here's our DNA here. And we're going to open it up, and we're going to start copying the information from DNA into messenger RNA. And so we drew this picture, and we labeled it. And so this particular enzyme does everything. So it's not like replication where there's two enzymes. There's only one in this situation. RNA polymerase is going to unwind and unzip DNA, and it's going to start building the mRNA. And we said that there were differences between the DNA and the mRNA in the sense that DNA has two strands. You can see that two strands. mRNA has only one. In addition to that, mRNA has U and no T. So that is another difference between DNA and RNA. So we're going to build this mRNA based off the information. And so we're only going to use one strand of DNA because, well, we can only use one strand because RNA is one-stranded. So we're only going to use one strand of DNA. Okay, and that strand is called the template. And the other strand that we don't use is called the coding strand. And that's transcription. So essentially all we're doing is we are copying the information in DNA into mRNA, okay? When that mRNA is built, there is an issue that arises because RNA polymerase is going to go through all of DNA and copy all of DNA. It's like taking a picture of something, right? Take a picture of something, you're going to take a picture of all of it. You're going to get all of it. The problem with that is that DNA has junk. So there is some junk in DNA. There's some parts of DNA that code for nothing. So the problem that arises is that those introns, the ones that the parts of DNA that code for nothing, get copied along with the exons, which are actual genes, actual expressed or coding DNA. So the junk gets copied too. It's like taking a picture of the whole thing. So the mRNA has pieces of junk in it. So if you're looking at this kind of orange part here, that's all the junk, all the crap that we don't want. Okay, so but RNA polymerase had no idea that it was junk, so it copied it all. So now what we need to do is we need to take the junk out and glue together the good stuff, okay? That's what we call mRNA splicing. So we're going to take this orange part out. Did we draw this last time? If we did not draw this, we did? Okay. If you did not draw this, you should be drawing it right now. We're going to take these orange parts out, the introns. So draw that now if you did not draw it last time. We're going to take the introns out because they're junk. And we're going to glue the exons together because that's the good stuff. And that's called mRNA splicing. I'll give you guys a minute to write that stuff down if you did not get that opportunity last time. So mRNA splicing. You have a primary transcript, the pre-mRNA, and then we're going to splice. I know it sounds like a weird word to splice something.
Is this for the part 2?
No, we're still on part 1, the back of part 1, the junk. So we're going to splice out or edit out or cut out the introns. And remember, the introns is the copy of the junk. So the junk stuff, the stuff that we don't want. So we're going to cut out the junk, and then we're going to glue together the exons to make that mature or that ending mRNA transcript. So let me repeat that so that everybody kind of understands. Remember, DNA has some parts of it that's junk that codes for nothing. It's just junk. Now, some people think that maybe that those junk nucleotides code for the soul. Who knows? Everybody's junk is different too, so that's interesting. Everybody has a different sequence of junk DNA, but the DNA has that junk that codes for absolutely nothing. Like it'll say just A or G, and it codes for nothing. But RNA polymerase, when it's going through transcription, doesn't know that's junk. So literally, well, not literally, but RNA polymerase is like taking a picture of all of the DNA, junk and all. Okay, it copies all of it, so that the mRNA that comes out of transcription has good stuff and junk. It has introns and exons. So now we need to cut out the introns. We need to get rid of the introns. And we need to glue together the exons, the good stuff. So make sure you draw that. Use different colors so that you can visualize it a little bit better. This is supposed to, I think this is supposed to be green there because the next picture is green. Okay. Are there any questions about that process? I don't want anyone to be confused. So once we have cut out all the junk in our mRNA and glued in all the good stuff, glued together all the good stuff, the stuff that we want, now we can leave the nucleus and start part 2. Wow. Okay, how we doing so far? All right, so now we're mRNA. We've cut out all the junk. We've glued together all the good stuff. Now we're ready to leave the nucleus and go out, find a ribosome, and start to actually build those proteins. Now, keep in mind, even though DNA gets a lot of credit, know that DNA is just instructions. It's like the instruction manual for building something. That's all it is. It's the proteins that are built by DNA or are built that actually go out and do all the work. Proteins do all of the work. Okay. So here is the junk, the intron, right? That's the junk. We cut out the junk. Notice it's cut out. And we glue together the good stuff. And now we're good. Okay, now we're done. I'm going to skip all this because you don't need to know it. And so we're going to go to the next. There we go. So if you would please go to part 2. mRNA is now perfect. It's been spliced and has all the appropriate or coding information that is necessary. And so now it leaves the nucleus. And now we're going to go out to the cytoplasm. And we're going to actually build the final product, which is the protein. Okay, so at the very top of your notes, mRNA leaves the nucleus and goes out to the ribosomes in the cytoplasm, and proteins are built from the instructions in mRNA. Please write that down. Say that one more time. No. Okay. So this is the part where I want you to go down to where it says translation practice. And I want you to write down, say that one more time. Okay, so the second part that says translation practice. And I want you to write this DNA molecule. And when you do, I want you to write it in sets of 3. That's really important. So please write them in sets of 3. Please write this DNA strand and please separate it in sets of 3. Once you do that, I want you to go ahead and transcribe it. Okay, so if I've got TAC what is the mRNA going to be? If it's TAC, mRNA is going to be what? A, U, G. And then if I have GCA, what's the mRNA going to be? C, G, U. And if I have CAT, what's the mRNA going to be? G, U, A. And then TTA, what's A, U. And then CGT is going to be GCA. And then ACG is going to be UGC. And the last CGG is going to be GCC. Now, this is stuff we already know. That's the mRNA. Now, we're going to start practicing trying to figure out what the amino acids are going to be. Now, remember I said that proteins are built of amino acids, like amino acid 1, then attached to amino acid 2, and so on and so forth. It's like a chain of amino acids. So bear that in mind. Once we have built this chain, we wrap it all up, and then the protein goes off and does whatever it is that it's supposed to do, whatever its job is. Okay, so now we're going to figure out how we use our codon charts. How to use a codon chart to figure out the amino acids. Okay? So this is a part where those of you guys who are at home, you can watch the little video. The rest of you guys, I need you to take out your codon chart that you got last class. It was kind of a funny looking chart. That's it. It says codon chart and wheel on top. So we're going to go through how it is that we figure out which amino acids to match with our mRNA code. For those of you at home, watch the video, and there will definitely be a codon chart available to you on your exam. So you don't have to worry about, well, am I going to have to memorize this? You don't have to memorize it. You will have a codon chart available to you. Same for you guys. You need your codon chart. Say that one more time. Yes, it. Does anyone not have a codon chart? Everyone has one. Okay, so it's my suggestion that you use pencil, because you're going to want to use your codon chart later. So you want to be able to erase any marks that you make. All right. It doesn't matter. I'm just saying anything that you can erase, because you're going to want to use it later. And if it's all messy, that won't be cool. Okay, don't get fuzzy on me, camera. All right. So the first set of mRNA nitrogen bases are AUG, correct? So I want you to label that nitrogen base 1. Oh, we went fuzzy. We went fuzzy. Come on. There we go. So this is your codon base 1, or letter 1, the first letter, right? First base, first letter. Here's second base, second letter. And then here's the third base, or third letter. Okay, so the bases are A, U, G, and C, or if it helps you remember, letters, right? So, so 1, 2, and 3. So we said A, U, G was first. So A, here's the first one, A, and then here's U. So I know that it's this row, and I know it's this column. Now the question that remains is which one of these 4 is my amino acid. So it's this row, this column. So the third letter, or third base, is G. So here's G. That's how I know which one specifically to choose. So A, then U, then G. So the amino acid is methionine. So underneath AUG, the easiest way to do this is just to write the first 3 letters underneath AUG. So it's met. So under where you wrote AUG, write met. All right, let's check out the next one. The next one was CGU. All right, so first letter C, yes. So we know it's this row, right? And then second letter is G, so we know it's this box. Okay. And the third letter, third base, is U. So we know it's this amino acid. So arginine. So just write the first 3 letters, ARG. Under where you see CGU. Let's do the next one.
Is this what like scientists use to figure out stuff like that?
Yeah, I mean, if you want to figure out which amino acid codes for which codon, yeah. Yeah. But the more you use this, the more likely you kind of memorize it. But you guys will never memorize. You won't use it enough to memorize it. All right, so GUA. So G, all right. Then we know it's this column, U, and then the way we figure out the amino acid, A. So it's valine. Okay, so AAU. So we know this is the first letter, right? A. A. So we know it's in this box, right? And then how do we figure out which one? U tells us exactly which one it is. And it's asparagine, so just put ASP under it. Okay, so you can use this one, so make sure you know. Is there any questions? Let me check and make sure, because I don't want anyone to be confused. Let me get 1 before I totally get this, Ms. Johnson, and 3, I'm totally lost. What the heck are you doing? And then 2 in the middle. Come on, fingers up. Let me see. 1, I totally get this. I totally get this. 3, I'm completely lost, and 2, I'm somewhere in the middle. Okay, let me do another one.
All right.
Which one are we on? GCA? Yeah? All right. So first letter is G, right? Yes? First letter is G. So we know it's this row. Second letter is C. So we know it's this column. So we know it's this box, right? And then the third letter is A. So we know specifically it is this amino acid. So you write ALA underneath GCA, ALA underneath GCA. Now the other one to use is this one. So you have a choice. There are tests that don't give you the choice, so that's why I teach both. So the STAR test may give you this one, or it may give you this one. Who knows which one it's going to give you. So make sure you know how to use both. So your next one is UGC, right? So you start in the middle. U, then you go out to the next one. G, and then you go out to the next one. C. So the answer is cysteine. So write CYS underneath it. And then the next one is GCC. So go to the big letter. This is the first letter, so G. And then the next big letter, C. And then the next one is C. So the amino acid is alanine. Okay, so this is what your paper should look like. So here were your mRNAs, right? And then you put the amino acids underneath. So let me see again. 1, I totally get this, and 3, I'm lost completely. Let me see your fingers. Let's go. Okay. All right, those of you on at home, we are switching back. Okay, so make sure you've divided up your mRNAs into threes, and those threes have a name. Can you please circle yours and write that? So every 3 mRNA bases is called a codon. Hence the name codon chart. Whoa, connecting the dots. Write that down. A codon is a block of 3 mRNA bases. Were you in the same spot, man? Right? Didn't you just do we did the translation practice, right? So in that area. I really do think folks decided to put it into threes because it's just so much easier to look at it that way than to look at it this way. That gets kind of confusing. So separating it into threes is just so much easier. Okay? So the reason why we focus so much on the mRNA is that first of all, we just copied it from DNA, right? That's the code that we care about, right? And that's the code that's going to be used to build the protein. So now this mRNA is going to go out to the cytoplasm and attach to ribosome. And when it attaches to ribosome, a tRNA is going to notice. Oh, let me get you guys to write this down before we move on. So in that same box, I'll tell you what I want you to write. Just know that the start codon is AUG. We're always going to start with AUG, which always codes for methionine. So every protein starts with the amino acid methionine. Every protein starts with amino acid methionine. And then there are stop codons. You don't have to write that down because it's obvious. Why? Because if you get to UAA, guess what? You're going to do what? Stop. If you get to UAG, you're going to stop. UGA, you're going to stop. It's really obvious. Okay, so I just need you to write down that the start codon is AUG. Okay. Now let's move on to the tRNA. So now the mRNA is attached to ribosome, and the tRNA finally sees it. And this is what a tRNA looks like. Go ahead and write this on the left-hand side. And then you're going to draw this one because the other one is insane to try to draw. When you draw it, please label the anticodon at the bottom. So the bottom of the tRNA molecule is an anticodon, and the top has an amino acid. Make sure you label that. The top has an amino acid. So the bottom has an anticodon, and the top has an amino acid. Okay, so the top has the amino acid. So I've got the anticodon at the bottom and the amino acid on top of the tRNA. Now, if I say the prefix anti, what are you thinking? Against. So opposite, yes? Okay. So anti means opposite. So therefore, anticodon is the opposite of the what? The codon. So the bottom of your tRNA has the anticodon that is opposite to the mRNA's codon. So after you write this information on the left-hand side, please draw this structure. Label the anticodon at the bottom and the amino acid on top. Now, next class, we're going to do some stuff with candy. Again, I got your candy for you. And we're going to do it together this time. Because I know last time, sometimes it gets kind of tough to do things by yourself. So we'll do it together. And we'll build a tRNA so that you guys kind of have a physical, tactile feeling for it.
Miss Johnson,
Yes, ma'am.
Which one are we drawing again?
This one right here.
Okay.
Okay, you guys ready? I don't want to go too fast, but are we ready? You need another minute? Angela, you here? Okay, Nikki? Is Nikki here? Nikki's not
I'm
here.
here.
She's online. Okay. Thank you. Isaac? Diego?
Here.
Hafiza? Is Hafiza here? No? Journey? Caleb? Elijah? Keston, are you online?
Yes.
Carlos?
Hello.
Keston's here?
Yeah.
Awesome. Carlos is here. Jose? Anna? Jamana's here. Jillian's here. Malik? You're here. Danny? Chastity's here. Katie's here. Jordan's here. All right. Awesome. Thank you. All right. Now let's go down to the ribosome. So what is the job of the ribosome? To facilitate the coupling of the tRNA to the mRNA. Now that is a really nice way of saying to stick the tRNA onto the mRNA. Okay, to make sure that the tRNA and the mRNA stick together. They have to bind. Okay? RNA is made up of ribosomal RNA. I'm sorry. Ribosomes are made of ribosomal RNA. Sorry about that. And that's kind of easy to remember because it's in the name. So ribosomal RNA makes up ribosome and proteins. There are two parts to the ribosome, a big part and a small part. Okay? And on the right side, go ahead and draw your ribosome. So next class, we will draw a ribosome. So you'll draw one big glob and attach a smaller glob underneath it. It kind of looks like a weird hat, I suppose. Bless you. Okay, let's flip over to the back, your part 2. So we're going to go through this process. The left side of the back of your notes is just transcription, right? So I just kind of drew a picture of transcription. That's what it looks like. That's what's going on there. And so now that mRNA has been transcribed and we've cut out all the introns, glued together the exons, now we can leave the nucleus, go out to the cytoplasm, and we can hook up with the ribosome. So let's start with that upper left-hand ribosome at the top. So you guys drew 4 ribosomes, right? And you wrote the word thin between each one. Okay, so we're going to start with that one that's in the upper left-hand corner. And we're going to start our process there. So the mRNA strand has decided to link up with the ribosome. I'm going to skip all this because you don't need to know all that. And we're going to do this real slow. Okay, so here we go. I want you to draw. So first, start by labeling your ribosome E, P, and A. Those are the 3 different sites on the ribosome. So label your ribosome E, P, and A. After you've done that, I want you to go ahead and draw your mRNA. I kind of started your drawing, and you can finish it. Label it mRNA, and go ahead and write in the codons. So the codons are AUG, CUG, AAU. Say that one more time. It's, yeah, on the right side. So some kids like to call it like a UFO looking thing. So in the upper left hand corner. After you've labeled your mRNA, go ahead and draw your tRNA. Now, remember when Ms. Johnson said that the mRNA has the codon and the tRNA has the anticodon? Look at that. The anticodon to the first codon is UAC. So see how those match? Make sure you write down the anticodon. Okay, so that is the anticodon. Okay, this is your tRNA. And do you remember what's on top of the tRNA? The amino acid. Okay, so the mRNA attaches to the ribosome and the tRNA attaches to the mRNA. Now, again, the way that the tRNA knows where to bind is it must find its match. Okay, so that if the mRNA codon is AUG, then the tRNA anticodon has to be the opposite of that. So UAC. Does everybody see that? Okay, that's how the tRNA knows where to bind. It's looking for that code that matches its code. Okay, so here's your tRNA, here's the anticodon here, and then the amino acid on top. Please draw that. Then draw the second tRNA and the arrow here. So we have a second tRNA coming along, and it's going to bind at the A site. Now, how does it know where to bind? How does it even know that it's binding in the right place? You guys tell me, how does this tRNA know that it's supposed to bind there? Very good. So the mRNA's codon is CUG. So the tRNA with the anticodon knows, well, I need to bind there. So the anticodon to CUG is GAC. So that's the tRNA that's going to bind there. So guys, this process is probably it's relatively simple, but if you're not focused, please don't distract yourself. Make sure that you are focused as you're drawing these, because if you're not focused, you're going to miss this. Okay, you guys ready to go to part 2, to step 2? All right, so heads up, I want you thinking. So first step was for a tRNA to bind at the first site, right? The tRNA knew where to bind because it matched its code. So its anti-codon matched the codon. Now a second tRNA comes and binds, and the way it knows where to bind is its anti-codon must match the mRNA codon. Okay, so here's your second picture that you're going to draw. So your next, the one that's after the word thin. Okay, so go ahead and draw that. So notice now it has the second tRNA molecule has bonded in the second site. Okay, God bless you. Please go ahead and draw that. It's the second one. So you should have 4 ribosomes. Everybody drew 4 ribosomes, right? We're on the second one. Go ahead. Okay, so now we've got two tRNAs, right? Bonded to the mRNA. And again, how does the tRNA know where to bind? Very good. So we've got this anti-codon here that matches with the codon there. Okay? Now, I want you to watch this. Heads up. What happened? Very good. The first amino acid bonds to the second amino acid. That's your third picture. The first amino acid bonds to the second amino acid. What is this bond called? Do you guys remember the bonds between amino acids? Ooh, we slept since, huh? That was a long time ago.
Peptide bond?
Perfect. There you go. Please label it peptide bond. Peptide bond. Now, question, do we need this tRNA anymore? Do you think we still need that? No, we're done with it. We can let it go. Okay, so we're done with that tRNA. It can go. It'll be recycled for another day. Okay, your last ribosome picture. We're going to let that tRNA go. And we're going to shift to the left. Did you see what I just did? We're shifting to the left. So we're letting this tRNA go, and we're going to shift to the left. Draw that last one. Now I want you to think. What do you think the next thing that's going to happen? What's the next thing that's going to happen? There's only so many steps. And you're on the last one. So what's next? We still have a codon here. Another tRNA comes along. This is my version of a tRNA because I can't draw. Another tRNA comes along and it binds here. Now question, we know that the anticodon's at the bottom, right? What would the anticodon for this new tRNA molecule be? UUA, because it's the opposite of the codon. So UUA. So another tRNA comes along with the anticodon UUA, and then on the top it's got some amino acid. We don't know which one, but it has an amino acid on top. And then we start this process all over again. This amino acid is going to attach to that amino acid. We're going to shift, and a new tRNA is going to come, and we're going to do it over and over and over again until we get a long string of amino acids.
Miss Johnson,
Yes, ma'am.
do we have to draw the first codon when
Yes.
in the last picture?
Yes. Okay, so in the end, we have built ourselves a protein made out of amino acids. Now, those amino acids all bonded together are going to fold all up, kind of clump all up, and become that protein, go out and do whatever it is that protein does. Now I want you to draw this all by itself. Just the sequence of amino acids here. All those amino acids bonded together. That's a protein. Now, we have another word for it because this is biology, so we have like 2 words for every concept, unfortunately. We have another word for it. What are those bonds called again? Peptide bonds. Okay, so we call this a poly. What does poly mean? If I say poly, what does that prefix mean? Like multiple. Okay, so polypeptide. So this is called a polypeptide because it has many, guess what? Peptide bonds. Or AKA just a protein. Okay, so polypeptide, also called a protein. That's what we've made at the end when we're all done. We've made a polypeptide or a protein. Next class, we will do this together, slowly, so you totally get this. Let's talk about homework. Now, last class, I told you we didn't get to where we needed to get to, and I said, hey, it's okay, you don't have homework. Okay, so here's what we're going to do this class. If you would like to do last class's homework, I'm going to make that an extra credit replacement grade just for this class. Okay, so say you have a 0 somewhere and you want to get rid of it, you can do that. But what you are absolutely going to do for regular credit for homework is the Schnapps activity. And that sounds so weird. I know. But you're going to do this activity. So what I want you to do is first you're going to click on that assignment and you're going to pick a monster. So heads up. So for homework, you're going to pick a monster. You got 4 monsters to pick from. Nobody cares which one you pick, 1 through 4. So pick a monster. After you pick a monster, you're going to go through and you're going to transcribe its DNA. So for instance, for monster number 2, the DNA is ACC. What is the mRNA codon? UGG. And then the next is GGT. So what is the mRNA codon? CCA. And the last is TAG. So the mRNA is AUC. Then you're going to figure out what the amino acids are by using your codon chart, right? So you're going to do that for all of the traits. Once you figure out the amino acid sequence, go back to the chart, look for the sequence, write down the traits. Once you've written down all the traits, draw your monster and turn it in. Questions?
So yours like left and yours like. Can we split it?
I prefer you didn't. Just write it on paper. Yes, ma'am.
So we only do like 1 of them, not all 3 of them?
No, yeah. Well, there's 4 of them. Just pick one out of the 4. That is homework. Like I said, if you want to do last time's homework for a replacement grade, go for it. Yes, Esther, they're both due next class. That's a good question. So yes, your homework is due next class. Now you guys had homework, not last time, but the time before. So hopefully you completed that. Tuesday.
And the extra credit is too?
Yes.
Okay, cool. It just says the salary piece, so it's okay.
It says what?
In salary, in Texas, it says due by today at 12.
Right. So it was supposed to be due. Yeah, you guys were supposed to already do it, but remember I excused it for you? But you can go ahead and do it and it will not be late.
But so, the assignment from last class was supposed to be done yesterday?
Right, but we didn't get to the information, so I excused it. And so now I'm saying you can do it for homework as an extra credit replacement assignment. But the Schnapps Monster is absolutely homework.
But, can I show you which questions that I think are like questionable?
Can I sit?
Huh?
Can I sit? Oh, hey, guys. By the way, your exam corrections are due by midnight tonight. So if you need to, are you guys listening? If you need to correct the last exam, that is due by midnight tonight. So make sure you get that done. Let me see it.
And then if Chelsea, do we ask her for the retest?
Yes, you may. That's fine, Keston. Huh? Say it again.
Do we ask her for the retest?
Yes. So after you finish your correction, make sure you text me or email me and let me know. And then I'll just open up your test so you can do it again.
Alright, so, like number 3 is like what part of sugar? What part of sugar is a part of the adrenal triphosphate?
Which sugar and part of amino diphosphate? Oh, you should have gotten that right.
And then there's some more too. But I had reminded you about some of them.
Okay. Find the other ones that are wrong.
I'll send it to you on remind.
You send it to me or mine? Okay, I'll look at it.
Send you like some evidence, like
Okay.
notes and stuff.
I'll look at that.
Oh yeah, could you please open the retake test, please?
Oh, yes, I will. I will do that during my conference.
Thank you.
Yeah, you guys can go ahead and leave. Class is over. Not you guys. Good try. Bye. Have a good weekend.
You got my retest, right?
Danny, right?
Yes.
Um,
I took the test yesterday. I'm just letting you know.
Thank you. Yeah, next time just email me or text me because campus doesn't tell us, unfortunately.